Hello everyone. Today we are going to study about classification of streptococci. So let's start with it belongs to domain bacteria, that is a single cell organism. Phylum is Bacillota or Firmicutes. We also call it Firmicutes. Basically, all the gram-positive bacteria falls under this phylum. There are some exceptions, but it is for gram-positive bacteria. Class is bacilli. Why we keep it under the class bacilli? Please do not get confused that streptococcus is a cocci, that means it's spherical in shape, then why it is placed in bacilli. And this class bacilli is usually of those organisms which have G plus C ratio less than 50% in their DNA. So this bacteria, which we are going to study in detail, belongs to a class which have G plus C ratio less than 50%. Moving on to the next slide, it belongs to order Bacillus, Lactobacillus or Bacillus. It's not visible, but order is Bacillus. So this order is that it can ferment lactose. So it belongs to Lactobacillus class. Family is Streptococcacia and genus is Streptococcus. Now, what are the characteristic features of this genus? This genus belongs to all I have told you, that's gram positive bacteria. Gram positive because when it's stained by gram staining, it looks purplish in color and cocci in shape, that means spherical in shape. It is arranged in chains. You can see the chains here in this diagram. So it is strap that is in chains and cocci is spherical in shape. This is gram positive. All the genus has this characteristic feature. It's a common cell in usually the species of this genus are common cell in human, and some of them are pathogenic also. So we will be reading in detail about the pathogenic organisms which are pathogenic to human. Then Streptococcus can be, genus Streptococcus can be further subdivided into obligate anaerobes. This is anaerobes or Peptostreptococci. Obligate means they are obliged to function in a particular way, and when it is obligate anaerobe, that means they cannot survive in oxygen. So they can only survive in absence of oxygen, while facultative anaerobes or aerobes can change their faculty and they can survive in absence of oxygen as well as in presence of oxygen. So they can change their faculty, that is why they are facultative anaerobes. Moving on to next slide, that is facultative anaerobes or aerobes are further classified on the basis of the hemolytic activity. That means they are grown on blood agar and what kind of hemolysis they are giving. So this can be divided into alpha, beta and gamma. Alpha is represented by a group. This Streptococcus viridans is a group which consists lot many species. So this alpha hemolytic is represented by Streptococcus viridans and Streptococcus pneumoniae belongs to this group, while beta hemolytic is represented by Streptococcus pyogenes and gamma hemolytic is represented by Streptococcus faecalis or Enterococci. Moving on to next slide, that is I just wanted you all to see what is alpha hemolysis. See this greenish discoloration, this is not complete hemolysis, it is partial hemolysis. So this rbcs show a greenish discoloration when the colonies are growing. You can see the greenish discoloration. So this is alpha hemolytic, that is streptococcus viridans. Beta hemolysis, you can see complete hemolysis, that means all red color is replaced by hemolytic colonies, which is giving a discoloration to the red color, and here you can see the colonies. And gamma hemolytic, no change, as the colonies are there, no hemolysis is produced, so you will see the similar color kind of, just you will see there is a distinction where colonies are growing but no discoloration has occurred. So this is gamma hemolytic streptococci. Moving on to next slide, we will study in detail the beta hemolytic streptococci because the most pathogenic organism belong to this group, and this most of the pathogenic organisms belong to this group. Beta hemolytic streptococci was classified by Lansfield, and this was a serological classification, that means in blood. It is noticed that on the nature of carbohydrate, that is the carbohydrate present on the cell wall of the bacteria. I'm going to show where this carbohydrate is present and what is its nature. On the basis of that chemical nature of this carbohydrate, that has been classified into 20 groups, where A to I and J are missing, otherwise all 20 letters are included. So majority of hemolytic streptococci that produce human infections belong to group A. So Lensfield classified the streptococci on the basis of the carbohydrate antigen present on the cell wall, and there were 20 different combinations. Out of 20 different combinations from A, we will start. I and J are missing, and group A belongs to the most pathogenic organisms are there, and in group A, streptococci is a representative species, or the group that represent group A is Streptococcus pyogenes. Moving on to the next slide, that is group A streptococcus pyogenes are also called gas, and this is further divided by Griffith into this Griffith typing was done, and on the basis of the presence of the antigen on the cell surface, what antigen is present on the cell surface, they have been classified into M, T, and R. So further classification of group A is this, and we are going to study group A Streptococcus pyogenes in the detail in next class. And here I have put a figure of bacteria where you can see this is the cell wall, and cell wall key basis, on the basis of cell wall carbohydrate antigen present, lens field divided into different types, and then Griffith divided on on the basis of the cell surface protein into MT and R, and we are going to study Streptococcus pyogenes in detail in the next. Thank you.